So let's see. So Nicole or Colleen, could you confirm that you can see the screen?
Yes.
Perfect. Thank you very much. So yeah, I'm super happy to present today because for me this presentation will be really helpful, especially the question and answer session. I'm a scientist, and as a scientist, it's always good to reach out to the community and really learn what are the topics and challenges at the moment, that we can adjust a little bit our research program and do really meaningful research, not just research which is done for research purposes. We want to do something which is helpful. So I will talk about epilepsy and genetics and maybe some words about who I am. So I'm leading here a research group at the Cleveland Clinic, and I'm affiliated with Case Western, but I also have a research group, a small one, like at the Broad Institute of Harvard and MIT, where I was before I came here. And I still have a small research group also in Cologne Center for Genomics. So by training, I'm not a medical doctor. It's very important. I'm not give clinical counseling directly to patients. However, my team is, and we are training medical doctors in how to look at genetic variants, and we are discovering actually epilepsy genes. So we're really at the forefront, and we are developing even software that medical professionals can make sense out of a genetic test, which is the biggest challenge so far. But this is not topic of today. Today, I will just give some background. I will start very simple. But this is always when a scientist says very simple is probably way too complicated, because I cannot talk simple anymore and talk overly complicated. But I will try my best. And then I will move up a little bit at the end towards more like specific epilepsy genetics, which might be a little bit more targeted for the healthcare professionals who join. But overall, I think we can get most of it if we just have a chat at the end. So please, I really encourage everyone to ask questions. There's no stupid question. And yeah, and looking forward to that. So before that, I will just give you some backgrounds and probably we'll start opening a lot of questions. So we'll talk a little bit about epilepsy genetics, what we know at the moment, and then also a little bit about genetic testing. And then I give a brief, like 5-minute outline what my research group actually is doing, because this is not part of the main talk I give at the moment. Okay, so if we talk about genetics, we talk about DNA. And to very simplify this topic and how you think about DNA, and actually, this is how I think about DNA. It's like a construction plan, and it's not as DNA is construction plan for everything. And keep in mind, every one of us originated from a single cell. That's pretty remarkable if you think about it. One single cell, half given from mom, half given from dad, makes the full thing we see here, right? It's an amazing construction plan, which enables more construction. Basically, it's the master plan. And if we talk about DNA, that's the master plan. And similar, I will always compare to a car. If you have like a construction plan like this for a car. So, and if we talk about genetics, we usually talk about genes, because all our diseases in genes. It's not somewhere else in the genome, because not everything in DNA is also a gene. Not everything, actually the minority is a gene. However, if people talk in context of disease, 99% of the time we talk about genes. And if we talk about epilepsy and genetics in disease, we talk about genes. There's nothing else. So, and if you think about a gene, it's just a piece of DNA, if you think about it, a strip, like a part of a strip, which encodes something. It's like a construction plan of a part, right? So I will, for example, refer to this in this talk as the part of a construction plan which encodes a break, right? So DNA, full plan, gene, part of it. So I already mentioned this a little bit, but I want to make this also a little bit more fun, that you also have to think a little bit. You might have had that in school, you might not. Depends where you went to school and when you went to school. You know, it's always a question, what part of the human DNA or the whole genome, that's all DNA humans have, is called genome, is actually making based off genes. So these essential building blocks. And it's actually like a small time apart. It's only 1.5% of our DNA. Until like a couple of years ago, we thought everything else would be junk. This was in the media also to some degree, but we learned better. So these other parts, they have some role, but we understand, which we in research understand more and more, but this is not clinically useful at the moment. It will very much become useful. There's already some evidence, or a lot of evidence for this in research. But if you talk to your epileptologist regarding, or neurologist or pediatrician or a medical doctor about genetic testing because of disease, they always talk about genes. And that's only a tiny fraction of our genome. So maybe, and also what might be informative, actually I will ask that later. So now I will give an example about how to think about an epilepsy gene, right? Just to make it also illustrative. So if we talk about a gene, that's a construction plan. So construction plan is important, but if the plan is not, you know, accurate or has some errors, then the problem at the end is not, for example, the car construction plan. That's why people die in a car crash or something. People die at the end because the thing which the construction plan is making or is encoding has a problem. And that's what we, for example, the brakes here. So the active molecule in the body which does something, which is producing something, which is putting something from A to B, or which does something, it's called protein. Okay, so the protein is the thing what is at the end having the problem which causes disease, and what people try to correct to some degree. So there are also some, you know, gene therapies, but overall people think about protein because that's the active thing in the body, like the brake in the car construction or in the car. So, and epilepsies have been in the past, still to some degree, that's the dominant type of, you know, genetic findings, have been thought about disorders of ion channels. So, and, you know, ion channels, you have to think about this as a tube, which is in your nerve cells, you know, in your brain cells. Our brain cells fire, that's their job. You know, they fire to communicate some information, you know, from, let's say, from here to here. So that's their job. And and if they can communicate from one cell to another, there are some chemicals go in and out, and they go through these tubes. So then these chemicals are ions in that case, and they go from, let's say, from outside the cell to inside of the cell through these kind of, you know, tubes which are, you know, in the borders between cells. And in the case of several epilepsy genes, here's an example. Here's SCN2A, that's one of them, but there are many others. It turns out that this gene encodes this ion channel. This is, and to make it more complicated, scientists give it a different name. So it's not, the gene is called SCN2A, and this is what people refer to in the clinical genetic test. But the active molecule, the ion channel, actually, it's called NaV1.2. So it's a sodium channel. So, and what you learn here now is, like, you have these now this channel, which is equivalent to the brakes, which, you know, also have this construction plan, right? So this is the active thing in the car, which is relevant for driving. And this is, you know, the active thing which is relevant that your neurons can communicate and fire. And here, that's a little bit more detail how that thing looks in a different schematic. Yeah, how it looks and how there's different functionalities. So, but it's actually not really relevant. So, and as I mentioned before, this kind of tube, this ion channel, is present on these many neurons. And this, for example, like a shot of neurons in a dish where you can see now how they are connected. And usually they would fire, let's say, from one direction to another direction, and would have, and the information, so the firing has to go from one cell to another. And these kind of, these tubes are between these cells. And you can imagine already, if one of these tubes is not working, there's a problem in communication, right? So think about it as traffic lights, right? So if one of these if this would be a traffic light on the highways between, neurons, if one of traffic light is malfunctioning and is always red, then you have a problem, right? Because then nothing is going forward. However, if this traffic light is always green or white, so it's always go time, that's also bad, because that can also lead to harmful situations. And this is a little bit how we think about epilepsy genetics. And if you understand this, and then you have understood 95%. We don't know much more, So it's we call it like dysregulation of excitation and inhibition. It's a fancy word for just traffic lights are off or on. So here's a slide, a little bit more detail. But I will also show this in a more simpler way. But for maybe our medical professionals. So every one of us has 2 genes, right? Every gene twice. So every gene we have twice, right? So we have one copy which we have gotten from our mom, and we have one copy from our dad. And under normal conditions, this is a little bit more detail, but I will explain it also a little bit more simple again. So in the normal conditions, we have each gene is producing like an RNA molecule. That's an intermediate step before you have the active molecule, like the break. And each of these genes does it. So you have one, more or less, you have, simply speaking, like 2 active molecules, and these RNAs become proteins. And this little thing here, this is how actually proteins are modeled at the moment. So that's fine. 2 genes, 2 proteins at the end, which can do the job. Fine. But there can be a situation where one of those genes has a mutation, and that can be inherited or can be de novo. So happens somehow very early in development. So when actually on the single cell stage, when the sperm and oocyte come together from mom and dad, suddenly there can happen a mutation. It's not really understood how that happens. There are some suggestions, and also there are some other ways, but in general, you can have a mutation, or it can be simply inherited with your dad or your mom has it as well. And then what can happen is that you don't have these, let's say, normal or you don't have the most frequent version of the construction plan. You have on one gene, you have a different version of the construction plan. Okay, doesn't need to be bad. But if this, mutation is affecting some essential part of the plan, then it can be bad. So, but we don't know how it can be bad. It can lead to many things, but I will talk about this. So, however, there are also other type of mutations which are weird. These type of mutations, you can have like a mutation which tells on the RNA level to destroy this piece. So which is degraded is the scientific word. In that case, this mutation you have here, it's a more bad one. We call it truncating variant. And this variant leads at the end to the situation that we have only one or like half of the active molecule in our body. So here in this context, we have still both, or the full amount of molecule, but one of them is modified, which can be fine or can be really bad. Or here in this case, we anyways have definitely half of it. So, and that's for many genes and many epilepsy genes bad, only having half of it. So there can be another situation where this gene is simply missing. You don't have it. So you only have it once from one parent, but you don't have it from the other one. In some situations where it is de novo, your parents have both copies, but you have only one, because at this very early stage, like single cell stage, there can be some kind of changes with your DNA, so your construction plan. Something is messed up because of unluck, and then you only have one copy of the gene, and that can also lead to the same situation, like you have like with this kind of mutations. You have both times only half of it, and that's bad. So, and there might be other situations where your DNA is also changed, and suddenly you have this gene instead twice, you have it 3 times, or even actually there are 4 times. So this is a big thing in cancer. In cancer, that happens a lot. And your tumors, the DNA is all over the place. But this can also happen to the basic DNA, your construction plan. And that suddenly in some individuals, sometimes de novo, so only in the affected individual and not in the parents, or sometimes it's also inherited, then you have too much gene at the end. So and too much gene leads also at the end to too much protein, which can also be bad. So just comparing this again more on the active molecule part. So you can have the situation, this is the normal situation, I would say, or the most frequent one at least. Normal is a dangerous word. So here we have, we have enough ion channel and it's like enough amount of breaks. So in this case, it would be like 2 breaks would be ideal for most cars. And in most genes, like 2 ion channels would be also ideal, just simplified speaking. However, you can have mutations which lead to the situation which you only have half of the amount of the ion channel, which is like having only 1 out of 2 breaks. Now that would also be bad, right? If you drive and your brakes don't, so there are not enough brakes. So there could also be a situation that you have a modified channel because of a mutation. And this can also be, imagine about brakes which are modified. If you drive a car which has super strong brakes and it doesn't start because the brakes are always on, it's very bad. And if you have a car where brakes don't work at all, that is also super bad. Or if you have a car where brakes work in a different way somehow, that can also be bad. And this is something which we face a lot so at the moment. And this is also one of the biggest challenges, because for these type of mutations, it's rather obvious what happens because you only have half of it because of a very clear signal we as geneticists can read in a clinical report. But here we always have to make a guess because the type of modification can be super bad, but we don't know how bad, if it's, if the brakes don't work at all or if the brakes work too strong. We cannot it's really hard to predict. And we can also not we start to distinguish between mutations which don't change the brakes at all. Because if to be honest, what I don't tell you now is, but I will tell you now, is that every one of us has tons of mutations, and I mean more than 10,000. So this means that, which partly lead to the situations that we don't look the same. So everyone looks a little bit different, but also but not all of these mutations do actually something. We have many mutations also in like epilepsy genes, but they don't do anything. So it's a big challenge for genetic counselors and medical doctors and geneticists at the moment, distinguishing between those which make a disease-causing change and those who don't. And then within those who make a disease-causing change, what kind of change they do. That is actually the biggest topic at the moment for genetics and epilepsy and genetic test interpretation. Okay, so this is an interesting slide. So because this slide I would present totally different, like even 2 years ago. One thing what you have to know is genetics is a super fast evolving topic. So 20 years ago, we had 2 epilepsy genes. 10 years ago, we had 10 epilepsy genes. 5 years ago, we have around about, 50 epilepsy genes, or 30 or something. And now it's like more than 100, and it's getting more and more and more. So it's not because the genes are suddenly, or people have more genetic epilepsies. The thing is, we didn't understand it before. And this was, if I would give the talk 3 years ago and I say, and the question would be why genetic testing, I would have said because of diagnostics. Because people figure out why they have the disorder, right? So it's for many very severe forms of epilepsy, there, as you know best, there's sometimes a long odyssey out to understand why, and the genetic test result can give an answer for that sometimes. So that would be my answer like a couple of years ago. Then like 2 or 3 years ago, I would say, hey, you get the diagnosis, but or you might get the diagnosis, but you also find a community of people who have the exact same defect like you. There are many foundations which are specialized on a single gene disorder. And suddenly you have biological twins, people who have the exact same construction plan default like you, which you was or your kid, and then they will also experience through their life the same problems your child has faced or might face in the future. So you can learn from this community a lot just by having the same denominator. It's because epilepsy, as you know best, is just a huge umbrella term. And it's probably more than 1,000 disorders which have some, groupings, but it's a big group of disorder. But for those who have a genetic defect, they are very similar to another. And this is also really relevant for thinking about treatment and management of patients. So that would be my answer like 2 or 3 years ago. So my answer today is why genetic testing? Yeah, because there are treatments and there's specific management. So this field of epilepsy genetics is evolving way faster than MRI, EEG, and MAC, SPEC, or other biomarker research. To be on top of genetics, you need to read every week papers. The forefront of research and where clinic begins in epilepsy genetics is very close to another, which is also a problem because some things are not well established. So you need really an expert to work with to really make a best clinically accurate decision because things are so close to each other. So yeah. So, and the good thing is now what we learned, because we sequence now a lot of kids all over the world, and we have big research programs, and also clinical genetic testing is in many institutions already standard, but reimbursement is still an issue. But still, genetic testing is more or less happening. And what we learned through that is that patients with different entities, and these are here now entities, just examples of entities by gene name, they share some similar biological problems or solutions in some cases. For example, these patients with mutations in this gene, they have a defect in a sugar transporter. So this gene encodes a sugar transporter. So again, like the brakes, part of, this gene encodes an active molecule, which is a sugar transporter to the brain. And what people have learned suddenly is that if you don't give these children with these mutations a specific treatment, which is ketogenic diet, these children will get severe developmental delay. So, and that starts around age 4 to 5 and will go on later on, like in life, and will become really profound, really bad developmental delay, and in addition to the epilepsy. And a simple solution is giving them ketogenic diet. Because if you do this early enough, then the child will be symptom-free. Literally symptom-free. No developmental delay, no epilepsy anymore. But if you do this at a later stage, when the brain has already developed under this suboptimal conditions, it seems that it's still beneficial to use ketogenic diet for these children with this genetic defect, but it's not so optimal anymore. So that's why I'm a big advocate for early on genetic testing. And also what you see here is these are rules which have just been established in the last 3 years, what kind of drugs to use. And still really a lot of development, which is very promising. And these are existing drugs here, what I list here, which are out and can be adjusted for patients with certain disorders. However, what you have to keep in mind is there's also something else we notice. These are children, for example, with the STM1A mutation. They have, 2 less sodium, basically, because of this mutation. This is because the ion channel, which brings sodium in, is made by this gene here. And the problem is, if you give these genes a sodium, these kids with these disorders a sodium channel blocker, and this disorder is Dravet syndrome, for example, you make it really worse because they have already less sodium, and then you put it on top of it, and then these children really suffer really, really, really bad. So this is an example, for example, where genetic testing can inform the information what not to do, not necessarily what to do. So, but he also know positive examples on which drugs are more beneficial. And there's a lot of research going on, actually, for this gene in particular. In addition to that, one thing to consider is that the information of the gene is frequently not enough. You need to know what kind of mutation you have. Because as I mentioned, I remember this example from the breaks. Imagine you have a mutation in the break. You need to know what does this mutation do, right? Because if you have a mutation which leads to the situation that your brakes are too strong, and then your car will not work, right? Just think about it, and then you will not have no firing in the brain. But if you have a mutation which makes the brakes not work at all, then you have too much firing, right? So your car would drive wrong. So these are two totally different problems which need different treatment. So you really have to not only understand what disorder, which gene, but you also need to understand what type of mutation did we found in the genetic test in that gene, so that informs how you treat or what kind of drug you need. So it's and this is an information we know since 1 or 2 years. So depends on the gene. So and this is it's very cutting edge. If you think about this, an MRI has been found like 30 years ago, 40 years ago, and there have been like 5 major types and so on in the last 40 years, and there's some software development. But here, genetics, everything goes through the roof in the last 10 years, and every half year there is a lot of new information. So I just want to stress that it's a very advanced, fast evolving field, and it takes a lot of energy to keep up. And that's why medical professionals need to have a genetic counselor who is specialized on epilepsy, or you need to have an epileptologist or neurologist who is really dedicated to keep up with genetics, and particularly the genetics of their disorder. I spent every day the last 7 years on epilepsy genetics and all genes. And I think we're giving international courses and train medical doctors in genetic test interpretation. I think we're quite good. But we are specialized in epilepsy. I could not give training in cancer genetics. And I would probably be able to do autism genetics, but I could not do Parkinson's genetics. So it's really important to understand, your topic, and because it's such a big field. Okay, so and even more hot, and this is, I think, really helpful and really makes many people hopeful. The next year, it makes many people hopeful, is that for many severe genetic disorders or severe genetic epilepsies, there is a lot of things happening in terms of gene therapy or gene modifying therapies, which is a hot topic at the moment in all the scientific meetings for epilepsy, because there are big pharmaceutical companies and biotech companies who show really good preliminary results in animals or in phase 1 clinical trials that they can basically modify the construction plan, so that they can really restore the disorder biologically, not just try to modify it with unspecific drugs, which is what's standard at the moment. But they really can, surgically go in, and it's a hope, at least, which looks really promising. There are lots of results, but they can really reconfigure the system with these specific gene-based drugs, and that might be really a solution for many patients with really severe genetic epilepsies. And if you just follow a gene therapy, ASO is a term for this, or targeted therapy. There have been so many biotech companies now supported with like hundreds of millions of dollars, which is amazing, to develop these therapies. And there's some good examples already in mouse where they could really remove the gene and then use this drug one time, and it really looks that the mouse doesn't have seizures anymore. It's really amazing. So that's why it's a really good time to be hopeful. And also, it's very important to do the genetic test now that we know, that we understand or find everyone who has these genetic disorders. Then when the clinical trials come in, they started already for some actually, but for other genetic disorders or other genetic epilepsy types, that we already have the cohorts, and then we can enroll these patients into the clinic. So it's very important to be hopeful, because a lot of things are happening. And this is amazing. So we're coming back to genetic testing. So we have a lot of different types of genetic testing, which has some historical background. These are all like specific types, and where in here you look at the whole chromosome, here you look at a single gene, here you look at more genes, here you look at all genes, and here you sequence the full construction plan. So it's basically screening. These are different levels of screening you can do. And so all this technology evolved so fast that these three, and hopefully in a few years only this one, which looks at everything, was clinically implemented. So again, few genes can identify, so these sequencing technologies can identify these kind of mutations, these exchanges, and this larger sequencing studies, or these these type of technologies, they can find these, genetic changes where you have too much or too less DNA. But even these, so these exome sequencing and panel sequencing can today also identify these kind of changes, although not so accurate actually, but they are improving. So, but at the end, the final solution will be the whole genome, because if you do whole genome sequencing, you have everything. So, but it's still in the US at least hard to reimburse. Other countries like Scandinavian countries are way more advanced in this. So, but it's a different healthcare system. Okay, so now we come to genetic testing in epilepsy. So, what is the role there? How many how many people are positive and so on. So, if you go to an epilepsy hospital, let's say, and this is a hospital which has old and young epilepsy patients, mild and severe, generalized and focal, and so on, and you just would make a guess. If you would, do a gene panel which looks at, let's say, the top 100 epilepsy genes, if you would sequence all epilepsy patients, how many would be genetic test positive? So how many would receive a diagnosis? So the underlying molecular reason why they have epilepsy through a genetic test. Okay, this is, okay, so it's not ideal, it's now, but it's 15%. So 15 to 20% based on literature, but I will go into this in more. So, and this is, again, a slide just to show you how clinical sequencing plays an increasing important role in epilepsy overall. So here are all studies which have ever been published in relationship to epilepsy. So the higher, the more, the larger the fraction, these medical terms have been more frequent, these medical terms have been used. So what you can see, for example, in the 1980s, around about 1 in 3 publications related to epilepsy used the word EEG as well. So because EEG is the #1 biomarker for epilepsy, at least, overall it is. And you can see that, for example, epilepsy surgery becomes more and more important over time, right? So more and more people write about it in research. And usually that's also like one step before clinical application, because a lot of research is clinical. So what you can see here, for example, MRI. So, they started here in the early 80s, and then became like a large part, increased tremendously within like 20 years. And you can see now 1 in 6 research studies has the word MRI in it. And it's when MRI and EEG are, part of the routine setup. And now I want to pay your attention to this part here. But you see that relatively speaking, these kind of biomarker, and here also a couple other ones, are going down. However, one thing which is really going up is genetics. So already now, around about 7% of all research studies have the word sequencing, so screening for genetic variants in it. So, and if you can just project it where we are like in 5 years or so, you can imagine that genetic testing is one of the most promising biomarkers for epilepsies overall. So, and the question still remains currently, what is currently the rate of individuals who are genetic test positive? And this is a study what Arto from our team has looked at, and we just try to answer this. So to be very brief, Arthur looked at the end at 80 studies from the literature, which we collected through a very standardized way, and this which have done sequencing in epilepsy and autism and intellectual disability patients. And this is the diagnostic yield. So it's, you know, what you can see here is that it's not a big difference, but you can see that around about 1 in 5 to individuals with any of these disorders will be genetically determined based on the genetics we know today, right? So that might even go up. So based on the genes we know today, in which we can screen, which we can see that around about 1 in 5 epilepsy patients will be genetic test positive. Just all epilepsy patients, even mild ones. But we know already that the severe patients with really bad epilepsy and really early onset, and also with, you know, cognitive problems, this type of epilepsy is the most genetic test positive. So, but still, 1 in 5. And this is a big number if you think about it, because this will explain the reason why that individual has seizures. This will be the foundation for targeted treatment which potentially is already existing, or treatment which potentially is better what is existing and might even be, since it's more targeted, leading to less side effects. But this is no more research, you know, but you saw already the slides before. So another thing what Arthur saw, and that has already been reported before in the literature, if you do the more comprehensive genetic test, you will get a higher positive rate. Meaning if you do just a panel, which is only looking at a few 50 to 150 genes on average, you might miss something. So if you ever in a situation to make a decision, should we do the more comprehensive test, which looks at all genes, or looking at a subset of epilepsy genes, do the comprehensive test. This is something which some insurances don't agree with, because the more comprehensive one is more expensive. But this changes because this technology is also getting way cheaper. So again, as a summary, you know, around about 1 in 5 patients, and this is unselected, who will receive, who does a genetic test will get positive. The more comprehensive test is more more successful than the less comprehensive one. And you know, we looked also globally, and we see that you know, genetic studies on genetic testing is increasing like crazy. So okay, so now I move on and just show you some how this is like a depiction of how many epilepsy genes we could find. Each of these letter codes is a gene, and you can see that it's increasing, and the slide is outdated. And today, or like now this is a little bit old. So we would think, depending on if epilepsy is the core problem or if there are other problems together with the seizures, we could count that we have, I think the true numbers today would be 50 to 150 genes. So, and this is changing. We have every month a new epilepsy gene to research. So if you do the comprehensive test, you can actually look back at your data and see if one of those new genes, which have not been evaluated previously, has a mutation. So that's why we would definitely do the comprehensive test. Work with a genetic counselor who can, or a geneticist or epileptologist who can re-evaluate this. Okay, so a little bit history. The first type of genetic variants were large regions in the genome, which were lost and could confer risk to epilepsy. So these genetic factors don't cause epilepsy, but if you have it, then you are 50 times more likely to get epilepsy. But these are relatively mild ones. So then we could identify some modifying genetic factors. These are relatively mild ones, which have been found many years, and you need many patients. But these are very genetic factors which many of us also carry. And individually, they don't do much. But if you have many of them, then they are conferring risk. But it's not, these type of genetic factors are not clinically relevant. And then we have these monogenic epilepsies, where if you have a mutation here, explains 100% of your epilepsy. That's the reason why you have it. And then there's a relatively new type of genetic factors, which is in patients who have some an abnormal MRI, so where you can see something in the MRI, so like a tumor, or where you can see so-called focal cortical dysplasia, like, or hippocampal sclerosis, regions where you see abnormalities in the brain. There we see like roundabout in 1 or 4 patients. If we genetically analyze the abnormal tissue, for example, some patients get surgery, and if you remove this tissue, like a tumor basically, and then you extract the DNA from this tissue, and then you look at this, 1 in 4 roundabout will have a genetic mutation, which explains why these cells look weird. And this is a new type of genetics. So it's similar to cancer, where you also have something abnormality growing in you, which contains a lot of mutations, but all other cells of your body don't have it. So this is something which you cannot see in the blood, because it's only in your abnormal brain cells. And this is a big research part also our group is involved in at the Cleveland Clinic. So genetics is very complex. There are some variants which definitely, we call these large effect variants if you have it. So variance is a mutation. There's some mutations, if you have them, that explains your epilepsy, 99%. So, and there's many genetic factors which give you risk to develop epilepsy. And that's you see in some families. In some families, you screen them genetically, you don't find anything, don't find anything in these monogenic epilepsies, but you still can see that in every generation, there's like 1 or 2 people out of 5 or so who have epilepsy. And you see that must be genetic, but we cannot point on it. And this is something which is also part of the research at the moment. And people try to discover this. And it looks that these kind of families or individuals carry thousands of tiny risk variants, which combined in some individuals surpass a threshold. So some individuals might have 70,000 and you get epilepsy risk mutations, where there's other ones have only 40,000 risk mutations and don't get it. And this is like a really hot topic in research. Okay, so as I mentioned, we have like around about 100 to 200 epilepsy genes, and it's really increasing at the moment. So one thing I want to point out for the medical professions, so just having the gene is not the solution, right? So it's not like we do a genetic test and then we have the results and easy results. It's not like this. So if you look at the largest database where all clinical results for epilepsy have been put, and also other disorders, and you look how people classify mutations, okay? How people, health professionals have classified mutations they find in a clinical genetic test. Most of them, they don't know what to do with them. They cannot tell, is this the reason why the patient has variants, has epilepsy or not? It's impossible to say. They cannot say these variants are benign, they don't do anything. They cannot say it's pathogenic. Why is that? Because we humans have so much mutations. And due to the situation is, if you do a genetic test and you look at many genes, you probably will get many genes which are positive for mutations. But positive for mutations doesn't mean pathogenic, because there can be many, if you think about the break, which I showed you at the beginning, there are regions in the break. If you have a loose screw at some regions which are in some regions, it might not be important. You might have a loose screw, but the brake still works perfectly. But if you have a loose screw at some parts where the brake, for example, grabs, I don't know, I'm not an expert, I'm not an engineer. So in some other parts of it might be really bad if you have a mutation there. So we are humans. So in every epilepsy gene, there are hundreds of mutations which don't do anything. So, and at the moment, we don't really have a map for, not a good map for every gene where mutations can be and where mutations cannot be. So this is, and this problem is reflected here. So you can see, you get a genetic test, you do a genetic test for like 100 genes, you end up with 4 genes, epilepsy genes, in your epilepsy patients, which are positive, and now you have to decide which gene is it. And this requires a lot of knowledge to move forward here. And again, you need to really have a lot of training to make an educated guess here. So you need to be able to qualify, is the gene on the test actually an epilepsy gene, because not all are, then you have to know if the gene fits really this patient's subtype of epilepsy. And there are more than 100 epilepsy genes out, so you need to know for every gene how the average epilepsy patient looks like. And then you need to know, something about the mutation itself, because I told you the mutation itself is very important. So, and what I sometimes observe is that some health professionals are not well trained, and they just do like a 5-minute review of the mutation because it's such a new topic, new gene, and then they try to come up with a solution, but then they at the end are not able to, right? So, and this is so that's what I try to stress throughout this talk the whole time, is that it needs really a lot of training and education of genetic counselors, specialized epileptologists, neurologists, pediatricians. So, and as a good example, for example, my research team and myself, we are sitting every week in the patient management conference where epilepsy patients get discussed at the Cleveland Clinic. And we're looking every week at EEGs since now 2.5 years. But I cannot read an EEG. I probably could learn it if I would spend a lot of energy. But although I'm sitting there and I see it every week, I'm unable to interpret an EEG meaningfully. So, and I should also not, because that's not my job. So what I want to communicate here, if for the health professionals, if you are looking at genetic test results, you need to do the training or involve someone who is specialized and trained, and it's a really fast-pacing field, to be able to make something out of the genetic test results. Don't trust just the companies, because the companies, as you and everyone is, also not perfect, and we need to, the field is changing a lot. So you have to be able to make a clinical evaluation yourself. And if you can do this, then you know that the gene, which gene fits a specific subtype of epilepsy, kind of clinical or like some genetic features associated with the genetic variants and inheritance, and then you can make at the end the decision. So it's rather complicated. Okay, I will just use 2 minutes to describe what our research is, and then we can start for questions, just to give a little bit shout out to my team and what research we are doing. So everything I just talked about was more like educational, but here what we're doing in research. So genetics is a little bit more complicated and it's already is used in the clinic. So, but I don't go much into this. I just want to show that our group is really involved in discovery of many epilepsy genes. We have been involved in the largest worldwide screens, and actually also even for some of those even executed the whole computational analysis and statistical analysis to identify those genes. We work globally with hundreds of clinicians together to not only identify those genes, but collect for every gene all clinical data from patients which are mutation positive, to understand what do all these patients who have mutations in this gene have in common, what they don't have with other epilepsy patients who don't have a mutation in that gene. So we call this genotype-phenotype correlations. And that's really helpful to understand what is really the specific problem with patients with mutations in this gene. Because you might have really different needs if you think about it, if you have mutations in a brake or you have mutations in the engine of a car, right? So it might be very different problems. So, and you need to learn this. Although at the end, your outcome is your car doesn't work, right? That would be epilepsy. But you really want to understand what kind of underlying problems are there. So then we also looking at different kind of epilepsy, these risk variants, which can modify and maybe explain some familiar forms of epilepsy. And this is where we are really pioneering a lot of research. Then we're trying to identify knowledge gaps, for example, how frequent disorders are and so on, how more genetic types of disorders are, how frequently they are positive, and how age plays a role and so on. And finally, we develop a lot of software. We are specialized on web software, which gives clinicians the tools in their hands to make the interpretations of the genetic test results. Or we work together with gene panel companies that they can do also the best genetic interpretation based on their ability. And these kind of research studies, they're not limited to epilepsy. They're basic human genetics and software development for all genetic disorders. Finally, we are specialized also on a couple epilepsy genes to use computational and informatic methods and biological methods and combine them all together with clinical data to make sense and move the field forward to understanding these disorders that other people who focus on drugs can really develop drugs with a lot of scientific knowledge. And all this would not be possible without a large team and a good supportive environment. And that's it. Thank you very much.
Thanks, Dr. Lale. I will give people just maybe a couple minutes here if anybody has any questions to submit. I don't know if anybody has submitted any questions to you directly. I don't have any.
No, I don't see anything.
Now. Okay, so let me we'll pause for a moment, and if anyone has any questions, now would be the time to submit those via the chat feature. In the meantime, while we're waiting, I will just talk briefly about some of our upcoming events and programs for the rest of the year. So this is only the second seminar of the year, so just be sure to take a look at the other seminars that we have coming up for the rest of the year. We have one on epilepsy and employment in August and one on caregiver burnout in October. We also have several other special events coming up, including our Purple Your Year initiatives, which help raise epilepsy awareness throughout the year. Our Race the River 10K, 5K, and 1 mile run on September 13th, and then our annual gala, which is on November 13th. So please visit our website for a full list of activities and other ways to get more involved with the agency. And then just a few notes on programming. We host an adult group that provides support and education for adults with epilepsy and their families. This group meets once every other month from 6 to 7:30 p.m. and our attendance is free. So our next meeting will be on June 10th. That will be held remotely. RSVPs are required, so you can give me a call at the office if you'd like to RSVP for that. And I did just want to share briefly that our adult case management program is open and accepting new clients at this time for telehealth services only. This is a program for adults with epilepsy and a mental health condition who are struggling to manage independently in the community. And you can reach me with any questions about this program. So it does look like we have a few questions that were submitted.
Okay, I can see them. Should I just...
Perfect.
Okay, there's one question is, if you didn't have seizures until adulthood, other than febrile seizures as a small child, could you still be positive for a genetic panel? So there's one rule of thumb, which is the more severe, the more early onset, and the epilepsy, and early, I mean younger than 4 years of age. And if you are challenged that you have problems in school, to really, at the end, that you even cannot go to school. These factors increase your likelihood that your genetic test is positive. So I think at the moment, if you have mild febrile seizures, there is one gene which comes into play, but it would not be clinically relevant, so just as a comment. And it's unlikely that you would be positive for that. But in any case, you know, for research purposes I'm very pro genetic testing in epilepsy, but I think it's very unlikely that someone who has a mild febrile seizure would be genetic test positive. The second question was that a lot of research is done in children. That's correct, when it comes to, especially when it comes to therapies and treatment. And how many adults are getting tested, or diagnosed? So at the end, every child will become an adult, right? So this is one thing what many adult neurologists forget. And traditionally, because genetics played in the past a bigger role in really congenital and early onset disorders, pediatric neurologists or pediatric geneticists are way more inclined to do genetic testing, just because of educational purposes and how it was done. Today, I would go again with these kind of discussions or features which I just described. If you have a patient which is an adult and that person had some, let's say, mild developmental delay or severe, or had an intractable epilepsy, so really the severe was everything which challenges life tremendously, I would do a genetic test, because here I don't know if no one has specifically looked only at adults. So the 1 of 5 rule would probably not apply. I would guess it's more like 1 in 10 or so. But you could be genetic test positive. And the thing is, this is all in the realm what we are studying at the moment. It might be that today we do a test which is negative, but if we look at it again with more knowledge in like 2-3 years, which is a long time in genetics, we might look at it in a different way. So to me, to be honest, I'm not a physician who has to deal with reimbursement. I'm a scientist. I would always do a genetic test. At the moment, many patients get only selected for genetic testing because someone feels or has the gestalt, that's a word for what clinicians use, sometimes that looks genetic, or that patient, that it must be genetic. And that's why we do the genetic test, and then we find out, okay, it's genetic. But all those individuals which have a mild version don't get sent to genetic testing, and because at the end there's no need for additional drugs, right? Usually the motivation is to find something which improves the condition of the patient. But if it comes to etiology and relate the reason why the patient has the epilepsy, I think we are learning at the moment also a lot in research that even for the severe genes, there are milder patients with milder forms, and you only see them if you do genetic testing. But I would agree that if you the least likely person to be genetic test positive with the current screens we do are individuals who have which are well functioning in society and have well-drugged epilepsies. That's the least likely group. However, many adult patients get epilepsy surgery because their epilepsy is not druggable, so pharmacoresistant, and this is because they have abnormal tissues in the brain. And these kind of epilepsies are also genetic, but then it's only this abnormal tissue which has. But here also genetics doesn't have yet a clinical implication there. I hope that was helpful. Okay, next questions. I cannot comment on with regards to allergies, but pharmacogenetics or genomics is an independent research or also clinical arm. So modern clinical facilities offer that you can do like a genetic test for adverse effects. And this is not directly related to, this is related to epilepsy, but it's more related to how you metabolize, or your body metabolizes drugs, or what kind of adverse effects you have. And a specialized physician can help you with this. So, but that's more indirect. Okay. Yeah, it sounds that the test is often not covered by insurance. There are initiatives, and there are companies like it's called Invitae Invitae, which have a program which is called Beyond the Seizure, where you can where a clinical provider can enroll you in a research study. I'm not a clinician, I'm a researcher. And here they can do genetic testing for free if you are younger than 7 years old. So for a kid, it works. Otherwise, the Cleveland Clinic is we under every academic institution is enrolling individuals for genetic studies. We have people at the clinic who can enroll individuals in our in-house research database. But it would not be diagnostic, so it would not report it back. That has some ethical or also regulatory things which do this. But I would encourage everyone to join these kind of studies, because as soon as we then find something, in genetics it becomes very fast, then a diagnostic test, and then it becomes clinically meaningful. And so in the future, then clinical session, you might get a clinical test for that thing we found because of you in research, and then that becomes useful. But it's a little bit more longer progress. Overall, academic institutions do research, and you are likely to be enrolled if you join these academic institutions. And yeah, that's the feedback. Okay.
All right.
Yeah, thank you.
So yeah, thanks, Dr. Lyle, for your presentation today. I think if that's all we have, we will be wrapping up. So I just want to thank everyone for joining today's online seminar. Hope you found the information helpful and informative. We will be sending out a brief email survey, so everyone just keep an eye out for that. And thanks again to everyone for joining us, and have a great day.
Thank you. Should I leave?
Colleen, do we need to stay on?
I think we're good.
Okay.
Okay, then have a good day.
Thank you, everyone. All right, bye-bye.
Thank you.